The first talk will be given by Nikolaus Rajewski. Nikolaus Rajewski is a theoretical physicist, a mathematician, and finally a biologist. And he has converted, I think, into a medical doctor also. For the sake of saving time, I'm not going any detailed introduction to him, except to say that he is now full professor for systems biology at the Max Delbrück Center for Molecular Medicine at the Charité in Berlin, here in Germany. He is scientific head of the Berlin Institute for Medical Systems Biology and received the very prestigious Leibniz Prize of the Deutsche Forschungsgemeinschaft, the German Research Foundation, this year. We are very honored, Nikolaus, that you're here, and we're looking forward to your talk.
Thanks very much for the nice introduction. And there's no timer here, but maybe you can give me a warning 5 minutes before I have to stop. So I'll talk about systems biology, and I'll give you a general introduction, just briefly, for the non-experts here, what systems biology is, and then I'll try to give you a specific example of how we try to implement systems biology here in Berlin. And finally, I'm a scientist, so I cannot not give you some science here. So this will be just 5 minutes, but about something very new, about which we are in the lab very excited. So systems biology, briefly, this is part of what happens in your metabolic system when you digest milk, sugar. This is just the chemical reactions which help to convert milk sugar eventually into energy that you can use. These are all reactions, and they follow certain rates, so this is a quantitative process. And of course, this is only part of the bigger metabolism, which is summarized in this slide. And I think from this it's clear, very clear, that this you cannot depict by symbols or descriptive language. You have to use mathematics in the end of the day to work with this kind of complicated system. And I think this is not only a lesson from studying metabolism, it's now, I think, an insight that is pervasive in biology, which is that biological function can oftentimes only be understood by quantitative models. So in the end, you have to map how the molecules talk to each other and make decisions about healthy cells, diseased cells, on a computer and mathematical models. And I think it's also fair to say that these models can only be obtained if you have enough data, and for this you need high throughput quantitative measurements. And I think what has happened in the past 10 years in particular is the development of such methods. And I don't have time to go into this, but a number of fundamental types of molecules can now be measured quite accurately, even in single cells. So I'm talking about DNA, about RNA, I'm talking about proteins. So a lot of this can be done genome-wide and quite accurately. So this makes what we call systems biology possible. There are now enough data to start building models and to try to really understand and map reality onto the computer. Now, you had a session today in particular where people discussed personalized medicine, or what is also called systems medicine, which I think is an incredibly important interface between the medical sciences and biology and all the interdisciplinary sciences attached to systems biology. So I think you can view systems biology as basically a fundamental science driving attempts to go into individual patients and individual organs or tumors of diseased patients. So for example, if you sequence the tumor of a particular cancer of a person, you can gain now, and there are now numerous examples, insight how to treat this individual patient. Okay? But this is only possible if you have some underlying understanding of what you see in this cancer really means. And for this, you need systems biology to understand this. So I think it's the fundamental underlying science for, in particular, multifactorial diseases such as cancer, cardiovascular, metabolic, and neurodegenerative diseases. And I mean, this is a very broad summit. I would like to say to the perhaps people interested in policies and politics here in the audience that I am convinced myself that if this is not happening, if this will not be implemented, then hospitals will simply be driven into obscurity and lose their grip, their funding, and patients coming to them. Because I think it's already now that if a person is diagnosed with cancer, he or she may already now try to go to a hospital where they offer sequencing of the tumor. If a hospital doesn't offer this, I think there's already now the possibility that the patient will not go there but elsewhere. So I think for larger hospitals, for example, the Charité here in Berlin, it's really the time to do something because this development is very quick. And personally, I think if nothing happens within the next couple of years, then within 5 to 10 years, these hospitals might really be struggling for survival. Okay, I'll maybe just say what I believe we need for medical systems biology to carry this out as research. We need basic research in disease models such as mouse and rat. I said we need high throughput technologies. I listed some of them here. And we need high throughput model systems which are amenable for high throughput perturbation and testing. So to knock out genes in a high throughput way to study exactly what happens when you do that. And there are some organisms, cheap organisms, which allow doing this. Now, systems biology by definition is highly interdisciplinary. And that's something that I cannot stress enough. You need mathematicians, computer science, biochemistry, molecular biology. You need medical research, engineering, and so on. So these people are not born like this. They have to be educated. They have to be made into being interested in systems biology. So I think what is absolutely crucial is to set in place modern interdisciplinary and actually global educational programs. I think this kind of science is not happening on one location or two or three. I think it's happening by really forming a network across the globe. And if you look at what some institutions like New York University or Harvard or MIT are doing, this is exactly what they're doing. They're going global with this kind of education. So I think in essence, another simple result of these considerations is that a modern systems biology institute should be equally balanced between computation and the wet lab, roughly speaking. And I think it's also fair to say that a traditional life science institute simply does not have this kind of setup. And so I'm very fortunate that in Berlin what we decided to do is, and that's really the success of many people involved, is to have a new building in downtown Berlin, which is on the campus of the Humboldt University. But this is a government, an institute funded mainly by the government, which will be built within the next 4 or 5 years or so, where we have the chance to really implement this kind of modern infrastructure from scratch. I think that's a huge advantage. I'm personally feel very fortunate to be part of this. And just some ideas about this. What we're doing is we have open lab where there's really no strict distinction between the computers and the pipette. So this is all a organic interface. Another thing is what you see here is that the principal investigators, the lead scientists, they don't govern over some lab, some giant lab. They sit next to each other so that we can really intensify the interdisciplinary communication. and many other ideas which I don't have time to go into. I think the concept is very attractive. We have been hiring across the globe, and I think this is developing quite nicely. But, okay, so regarding education, what we have built, and I'm just presenting this here because I think it's just an important thing to realize. You cannot on one place educate this next generation of systems biologists or system medicine people. You have to connect to other places. You cannot do everything in one place. So what we are building is, what we have built is a PhD exchange program with NYU, and I wrote the basic facts here. Here are really the most important thing, the people, the students currently in the program, and we are now opening up this program to other cities on the planet. Now, this was all about some general thoughts about systems biology, and I hope I have like 7 minutes left, Mr. Chairman, because, as I said, I'm a scientist. I'm not a manager, really. And so I decided that I would like to present you a piece of science which comes out of a lab, in fact, my own lab, where we work both with the computer and with biochemistry and molecular biology. And I'll just try to explain this. Even if you know nothing about molecular biology, you'll maybe get some idea of what I believe is the power of this approach. So what I need to tell you is that in your genome, the DNA, your hereditary material, has to be active. The genes there, which code for a protein, have to be transcribed into RNA. So here you see an RNA molecule, a messenger RNA. And this message has to be translated into a protein. So down here is a product of the gene, the protein. Now, the decision to make a protein is a complex one, and there are many proteins actually from other genes involved in regulating this. So you want to be sure that you really want to turn on activation, production of the protein, both when making RNA and also when making protein. Now, for the more experts here, for molecule discovered roughly 10 years ago, microRNAs are very interesting. These are not proteins, but small pieces of RNA which bind to RNA molecule, and by doing so, they regulate how much protein is coming out of the RNA. So it's a regulatory mechanism, okay? Now, a question that for 10 years is out there, which is a fundamental question for life, is how can the cell actually get rid of microRNAs? Once they're bound, then this messenger RNA is always bound and regulated by this microRNA, but that is oftentimes at some point not wanted anymore. Also for therapeutics, you want to interfere with gene regulation, so you want to break this interaction, or you want to somehow get rid of these microRNAs. So it's also for medical purposes, so this is a fundamental question. And one idea that is out there is that what you can use to accomplish this is to introduce a sponge. What can a sponge do? A sponge can take up things, and just imagine that this sponge is such that it can take up microRNAs. So if you would introduce such a sponge in a cell, then it would take up the microRNAs, and it would therefore sequester away the microRNAs from their normal targets, and therefore you would clear the cell from the activity of this microRNA. So what we did is we performed search, and so we searched actually a huge amount of biochemical and other types of data to identify this spot in the human genome where we discovered that this piece of RNA, which is made by this piece of human DNA, contains 74 binding sites for a certain microRNA. So this is very unusual. These sites are actually highly conserved, so they're likely to be functional. Now, the beauty is that, and this was just a coincidence, that it is known that this piece of RNA is actually covalently closed. This is something that people think is very unusual. So in textbooks everywhere, when you read about RNA, you're talking about linear molecules. They have a beginning and an end. They have a head and a tail, a 5' end and a 3' end. This RNA doesn't have such a thing. It is just closed. It is covalently closed, actually by the splice machinery. So it's a circular piece of RNA. Now, okay, so I don't maybe have time to go into this, but what we showed is that really this thing acts like a sponge for a microRNA. So in mouse brain, you see that the microRNA and this sponge I just showed you are co-localized. And in zebrafish, when you knock down the microRNA or you introduce a sponge, you should get the same phenotype if really the sponge is a sponge. You see that in developing animals, the forebrain is normal, but the midbrain is very significantly shrunk in size and sometimes actually completely obliterated. So this sponge is somehow regulating midbrain size. And so that's a whole interesting story on its own. But what I really want to tell you is that we, of course, asked, are these circular RNAs, is that an exception, or are there maybe many more? So that was a question. And driven by this, and I'm making this point, you see, this was not foreseeable. This is not plannable science. This is nothing you can possibly write on a grant. You want to discover something like this. This comes because there's a certain infrastructure in place and people who can read the computer and the cell. So the question was, how many circular RNAs are there in human cells? And we approached this problem by a computational one. So you see, you can sequence RNA nowadays quite easily and in huge amounts. You heard about this morning. And if you think about it, normal splicing in the cell is linear. So a piece of RNA can be ripped apart like this, just in the direction of head and tail. But for a circular RNA, this is vice versa. So the head becomes the back and vice versa. So we looked at many of these reads and by a computational approach, sifted them for cases where we have the indication of a circular RNA. And here's the result. In humans, we find 500 plus different circular RNAs. They are tissue specific. We also found them in other animals where we looked at developmental expression, so they're also stage specific. There are actually many of them reside in non-coding RNAs, but there's also many which are encoding. And the most important thing is, I don't have time at all to go into this, but we very carefully validated them. And just to say that most of what we, from our computation, the cases we looked at, we could validate. So what you have here, okay, I don't have time to go over this, but this is my summary. What you have here is, first of all, we identified a non-coding circular RNA that functions as a sponge to take up a microRNA. That's actually the first known function of a circular RNA. Second, we found a novel class of regulators in the cell. Okay? So nobody has ever considered these circular RNAs to be so broadly expressed, so specifically expressed, and actually also to be so conserved, nor have they been considered to be acting as regulators and having a regulatory impact on other genes. And so we also think that they have interesting application as biomarkers because they're extremely stable. You see, since they're circular, they cannot be attacked at the ends, which is normal for RNA to be degraded eventually because enzymes go and attack the head and the tail. But they don't have such things, so they are very stable. So I think they are interesting biomarkers and therapeutical agents to interfere with microRNAs. So in summary, necessary for the success of this project was asking an interesting question. You cannot just create a lot of data. You somehow need to ask a question. You need quick access to an enormous amount of data generated, in our case, in-house and worldwide. You need high compute power and an interdisciplinary team. Here's actually the team, the young people doing this, mostly PhD students. That was before we understood what was going on, and this was after. And with this, I'm done. These are in more detail all the people. I would also like to thank the Max Delbrück Center, where we have a very nice systems biology initiative. Thanks for your attention.
Thank you very much. I think we could have listened to the first part and the second part much longer. Let me just ask one question with regard to the first part, the more general part in system biology. So what you say, education, of course, is global. However, the science is very local. And you have designed a lab where wet chemistry and biochemistry is combined with computational work. What you need, however, in addition, is clinical work. How do you integrate that in your model?
Yeah. So what I try to explain is that so the part systems biology, what I described, I see as a fundamental science driving, I think, what is called now systems medicine, which is really then taking these approaches into the patient, sequencing cancers and so on and so forth. But understanding then from this point what's going on requires systems biology. So really what we need is, as you I think indicate, is proximity to a clinic and to medical science. And this in Berlin is actually a wonderful development, because this is exactly developing. I don't have time to go into this, but this is, I think, happening very nicely. Has a high chance of that Berlin becomes a primary point here in Europe for this.
All it needs at the end is good people. So thank you very much.